My name is Patrick Haggard. I work at University College London, and I'm one of the investigators on the Touchless project. And I've designed a talk which is really a tutorial introduction to tactile sensing in humans. And it's targeted to people who, for example, are coming from the engineering side or coming from the user interaction side, and who may not have read much about the tactile receptors, the tactile neurophysiology, and the tactile perception from neuroscience and psychophysics. So it's, for some of you, you'll know all of this already, but for some of you, I hope it's a useful introduction. And I'm going to try to do the following 5 things. I'm going to talk about the different classes of cutaneous mechanoreceptors. That just means the specialized cells in the skin that turn mechanical energy into electrical impulses in neurons. I'm going to talk about some other receptors which are relevant for touch. I'm going to talk about how the signals from those receptors reach the brain through the somatosensory pathway. I'm going to talk about how our mind or our brain produces our perception of touch from those inputs. And then I'll talk a bit about active touch. Okay, so all textbook discussions of neurophysiology of touch begin with the 4 main classes of cutaneous mechanoreceptors. These are the really important basis for a lot of our sense of touch, not all, but a lot of it. So a really good principle in studying any sensory system is to always start with the stimulus and to be aware of what your stimulus is, because what your stimulus will do is it will stimulate some receptors. The receptors are the cells in your body which convert the energy in your stimulus into electrical impulses in your nerves. And of course, there are lots of different types of tactile stimulus which we can classify: single point, multi-point, static, or vibrating. These are vibrating. These are motion stimuli which sweep across the skin, at least over in a very small local area, and can deliver an experience of texture. Now, these different kinds of stimuli will all have different effects on the receptors. So the important point to remember is that these stimuli can be beautifully designed and engineered, and they can be quantified, but they're quantified at the skin surface. And the way that the stimulus propagates through the skin tissue is generally not known. So we know what's happening at the point that these von Frey wires touch the skin, but we don't know how that's propagated through to the receptors. So the effective stimulation receptors is therefore unknown. And in particular, nonlinear mechanical interactions within the skin are likely, particularly for complex multi-touch stimulations. So something like this tactile array, where you can have multiple stimulators all touching the fingertip at a time, you're likely to get interactions. So what are the receptors that these stimuli are going to touch? Well, here is a cross-section through the skin of the fingertip. This kind of diagram is in all of the medical textbooks. Here's the surface of the skin. You can see the ridges of the fingerprint, and you can see the different layers of the skin, the epidermis and the dermis. And there are 4 main classes of cutaneous mechanoreceptor: the Meissner, the Merkel, the Pacini, and the Ruffini, right? So I'll tell you what those all do and what they all mean, but they're also associated with these sort of abbreviations, which I'll explain on the next slide. You'll notice that these are all encapsulated. So here is the actual nerve leaving the capsule, okay? But they're an encapsulated ending. And importantly, they can be either superficial, close to the skin surface, like the Meissner and the Merkel, or they can be deep, like the Pacini and the Ruffini. So the skin is a layered structure, and some of the receptors are deep within the skin and others are right on the surface, and that's important. We'll come back to these guys, these free nerve endings, maybe later. Now, so this is another rather similar cross-section. And do you remember I said there were 4 main classes? And the 4 main classes can be divided according to 2 crucial features of how they turn mechanical events on the skin surface into electrical impulses in the nerve. So first of all, we can divide the receptors into fast adapting versus slowly adapting. And that's shown here in C. Here at the bottom, you have a stimulus, which is pushed into the skin, stays there, and is then released. So this is like an indentation, as if you were to press against the skin of the fingertip with a stick. And the fast adapting units tend to respond with a spike. This is a recording from the neuron, which sends signals from the receptor to the central nervous system and the brain. The fast adapting units respond with a spike when there's a change in the indentation of the skin, but they don't respond during steady indentation. The slowly adapting units respond during steady indentation. They still adapt because they respond more when the indentation is changing than when the indentation is constant, but they at least do respond when the indentation is constant. So that's what FA and SA refer to. And the second important classification is that the type 1 receptors, the FA1 and the SA1, have small receptive fields. That means that this neuron will only respond when you press in a quite small localized region of the skin, okay? And the two types have much larger receptive fields. They will respond when you press more or less anywhere on the fingertip, all right? We've already seen that there's a skin depth effect because the type 1 receptors are superficial and the type 2 receptors are deep. And there's also a really very marked variation in how many of these receptors there are per area of skin. So if you look at the hand, for example, the FA1, which corresponds to the Meissner, is very present. There are many of them, 120 per centimeter squared on the fingertip and rather few on the palm. The SA1 also fingertip, many on the fingertip, but not many on the palm. But the type 2 receptors are actually pretty equally distributed over most of the hand and in fact over most of the body as well. So what this means is that the high sensitivity of the fingertips, the exquisite sense of touch that we have on our fingertips and in other areas of our body, like around the mouth, is really due to the large number of FA1 and SA1 type receptors that you have. Now, you remember on the previous slide, I talked about Meissner, Merkel, Pacini, Ruffini. Now you can see that the Meissner is the ending that corresponds to the FA1 neuron, FA1 afferent neuron, and the Merkel, the SA1 afferent neuron, and so on and so forth. Now, a lot of what we know about these receptors comes from a beautiful technique called microneurography. So in microneurography, a very, very thin wire electrode is inserted through the skin, and it actually is placed into the bundle of nerve fibers that run from the skin towards the central nervous system. So here, the mechanoreceptors on the fingertip that we previously were looking at, they send their afferents down the median nerve, and there's a particular point in the forearm where we can guess where the median nerve is, we can insert the electrode, and we can record from a single nerve fiber. And with the microneurographic electrode, you can record from a single nerve fiber, you can touch different parts of the hand, and you'll suddenly find that the nerve fiber will begin to fire. You'll be able to record its electrical impulses. And that means you found the receptive field. You found the particular point on the skin which overlies the receptor that the afferent fiber that you have got your electrode in projects down. And you get very similar kinds of recordings microneurographically to what I've just shown you. So here is the indentation of the skin in gray, and here is the response of a slowly adapting neuron, a rapidly adapting neuron, and a Pacinian corpuscle. In these upper plots, a single sort of tick, a single line, a single row is a trial, one sort of recording, and each tick is an action potential or electrical impulse. And you can see that the SA1 fires during the sustained response, the sustained pressure, the RA1 fires at the onset and the offset, and the pacinian really just fires at the transient. It just fires when there's a sudden change in the skin in the skin pressure. And you'll remember the pacinian is a deep receptor. So probably what's happening is that when you touch the skin, you produce shock waves that propagate down into deep layers and trigger the pacinian. Now, each of these mechanoreceptors responds to a distinct mechanical frequency. That's why, for example, you can see that the slowly adapting one, which is the Merkel, has a sustained DC response, the RA sort of slightly higher frequencies, and the PC very high frequencies. So here is frequency on a log scale of a vibrating stimulus, which is pushed against the fingertip. And here is the a measure of the sensitivity, effectively, of the firing rate of a SA type and an RA type neuron, and here a PC type, pacinian corpuscle type neuron. And you can see that basically the receptors and their afferent neurons are tuned to different frequencies, so that the whole frequency range from steady vibration through to steady pressure through to fast vibration is covered. And this is the reason why, if you have been sitting on your bottom all day, like I have, you can still feel the pressure on your bottom using the slowly adapting mechanoreceptors. But if you have something which gets inside your clothing, like a wasp, which is fluttering its wings at very, very high frequency, you'll feel the buzz of the wasp inside your trousers. So the whole frequency range is covered. And this has led to what one might call the filter bank model of skin sensing. So here is, in fact, the cochlea of the ear, right? And we know that the membrane of the cochlea projects different frequencies of sound to different points along the basilar membrane, and auditory nerves hang off this membrane at different points. And that's how you can hear all the way from high frequency to low frequency. So that's an auditory filter bank. And the skin basically has a pretty similar kind of frequency filter bank according to classical models, because you have different receptor classes which tap different frequencies of skin vibration. And some beautiful work by Saal and Ben Smyer basically produces quantitative models of the different kinds of slowly adapting, rapidly adapting, and PC afferents, simulates stimuli that are pressed against the skin, uses transfer functions and integrate and fire neural models to predict what a given stimulus is actually going to produce in terms of electrical impulses sent from the skin to the brain. And this is now the dominant approach in tactile cortical neurophysiology. It's a time domain approach. It's resolved according to the frequency preferences of the different receptor classes, and it's got some actually quite powerful transfer function models. So you can pretty much model what's going to reach the brain for any given tactile stimulus. Now, those are the classical receptors. But I think it's really, really important to mention what I've called the auxiliary receptors. This is not a proper term. This is a term that I use. And the reason that I use it is because many studies of tactile perception, in quotes, actually involve multiple non-mechanoreceptor sources. So outside of neurophysiology, a lot of people will talk about touch, and they won't mean the 4 classic mechanoreceptor classes. They'll mean loads of other things. So, for example, people often in user interface world, they'll talk about thermal sensations as being part of touch. Now, the thermal sensations actually come from a separate set of receptors in the skin, which don't respond to mechanical energy at all, but only respond to thermal energy, either in the warm or in the cold range. Many stimuli on the skin will activate pain receptors, even at quite low pressures. So a lot of what we call, what we might call touch perception could include signals in pain receptors. And possibly actually the most important one is the signals from proprioception and kinesthesis. So these are both quite sort of strange terms, but what they really refer to is some sensory endings, not in the skin at all, but in deep tissues, particularly muscles, but also tendons and joints, which are very, very relevant to how we use touch for perception. So first of all, let's start with the receptor. It's always good to start with the receptor. So this is actually a muscle. This is nothing to do with the skin at all. And inside the muscle, you have fibers that contract, and you have other fibers called spindles, which can't be contracted voluntarily, but which are actually sensory fibers. They're not motor fibers, they're sensory fibers, and they have a sensory ending wrapped around them. So that when the muscle is stretched, a signal is sent from the muscle to the brain saying, I am being stretched. Now, of course, muscles don't stretch by themselves. Muscles can only contract. But when a muscle contracts, it will always stretch its counterpart muscle on the opposite side of the joint. So if you look at your arm, you've got a biceps here and a triceps here. So if I contract my biceps, which I'm not very good at, I'm stretching my triceps, and signals are going to be sent from my triceps to my brain to say, I am stretching. Now, these are really important because if you take a classic touch perception task, here's a beautiful one by Astrid Kappers, where there are all these funny shaped objects, and they're hanging on a string, and you just grab them. And your job is to say something like, you know, are they round or are they square? Or your job is to say, how many triangles are there in this, what she calls haptic glance. Now, the point about this is that this is a tactile task. There is stimulation of the skin, but when you grab a bunch of objects in your hand, there's also contraction of muscles. And as you touch the objects, the muscles that are antagonistic to the ones that you're actively contracting will get stretched. And the way that they get stretched will depend on whether you're grasping a cube or whether you're grasping a circle. So whenever we interact with an object using our hands or any other part of our body, we're always going to have motor movements which will have sensory consequences for muscles. And this is the classic work by Lederman and Klatsky, where you're haptically identifying different objects by playing them with your hands. So the point is that shape and force information in tactile perception depend on a combination of tactile inputs from the skin, that's what we talked about before, that's mechanoreception, cutaneous mechanoreception, and proprioceptive and motor information, including motor information from muscle spindles. So just to give an example of the real importance of this auxiliary proprioceptive or kinesthetic signal, here's a really nice study by Vincent Hayward and colleagues. So what you're doing is you've got your finger on a little kind of railway train, and you can slide the railway train backwards and forwards on a surface by making movements like this, okay? And the railway train's under your finger. It's a little bit like a computer mouse, really. The mouse, let's call it a mouse, the mouse might go over a bump. So on the surface, they can put a bump. And of course, you feel that. If you're using a mouse and the mouse goes over a bump, you'll feel the bump in the surface. And you'll feel it in two ways. You'll feel it partly as a tactile sensation through your finger, but you'll also feel it because the bump will slow down the side-to-side motion of the mouse. So what Vincent Hayward did very interestingly was he separated these two components. So here's a physical bump and a physical hole in the surface. And what Vincent Hayward did is he controlled the friction in the mouse wheel that you were moving across the surface so that he could produce a resisting force followed by an assistive force that defined a virtual bump, and an assistive force followed by a resistive force that defined a virtual hole. And what I'm showing here is the probability of the person saying that they had moved over a bump. And if they're moving on a flat surface, they never think they're moving over a bump. If they actually move over a physical bump, then they say, yes, I moved over a bump. But if they move over a virtual bump, which is in fact not a bump at all, but just a change in the resistance to their movement caused by an increase in friction and therefore sensed as a change in the stretch on the muscles, then they say it's a bump. So the point here is the illusion of a bump is created by resistance, and you've got tactile sensation without mechanoreception or without cutaneous mechanoreception. And this I really want to emphasize this because it shows that there's a really strong so-called kinesthetic or muscular contribution, particularly in any kind of tactile task which involves naturalistic haptic movement. Okay, I put this in as an auxiliary sensation, but of course it is actual mechanoreception, is so-called affective touch. So in addition to the 4 classic mechanoreceptors, there's another kind of tactile receptor that was recently discovered. And we don't quite know what the receptor is, but we do know from neurographic recording that there's a set of afferent neurons which receive from the skin, and they're unusual because they have very large receptive fields. They'll respond to stimulation on a large region of the skin, and they are very slow responses. And the reason they're slow is that they've got they're unmyelinated fibers. They're slowly conducting. So if you, here is a stroking movement applied across the skin here, and you can see that it's done at 3 different force levels. And these neurons, each spike here is a response from the neuron. They care a little bit about force. So you get slightly higher spiking rates for the higher force, but they don't care that much. And they respond long after the stimulus has actually passed over the skin. And that's because these neurons take a long time to conduct the electrical impulse up to the recording electrode. But crucially, they show many more, these neurons have a strong preference for slow stroking velocities. And that's why they're thought to be involved in this kind of social, pleasant touch, like cuddling, grooming, stroking, and if you're a monkey, picking up fleas. So this is just the number of these units that you can record as this is the key graph. This is people rating how pleasant they find a stroking stimulus on their forearm like this as a function of the velocity. And you can see in the dashed line, the dashed line is the normal data. You can see that people prefer to be stroked at about 3 centimeters per second, at about 3 centimeters per second. That's pleasant. It's less pleasant stroking more slowly and more rapidly. And the solid line, interestingly, is a set of participants who report that nobody ever touches them. I don't know who they are, whether they're people who are old or kind of miserable, or I don't know, something like that. But these are people who say that they're deprived of touch, and they don't show this response to pleasant touch. So these are really important. These are probably very important for sort of social and affective touch. They're probably less important for perception, and you only find them in your hairy skin. You don't find them in the fingertips. So you don't use these to when you're typing, but you do use them when you're cuddling somebody. Okay. Right, so we've dealt then with the receptors and with the neurons that immediately receive from them. And then all of these signals go into the spinal cord through the dorsal root, ascend through the spinal cord, cross over in the brainstem, and go up in a big pathway called the medial lemniscus, and they go up to the brain or the cortex. And the main region that they project to is the somatosensory cortex on the opposite side of the body. So you'll be familiar with the so-called Penfield homunculus, the map of the body surface that you can see on the somatosensory cortex. You'll be familiar with the fact that there's much more of our brain devoted to skin input from the finger and the face than, say, the feet or the trunk. And that's because there are many, many more receptors in our fingers than in our arms or our trunk. So this is a sort of classic image. But what I want to talk about when I'm talking about the pathway is that classically people think about this as just being a wire going all the way from the skin to the brain. But in fact, there are lots of interactions between sensory channels. And I'm going to concentrate just on the interactions that occur in the spinal cord. So here's a slide. Here's some slides from a beautiful study by Abrera and Ginty, which show the inputs coming in to the spinal cord. These are the different layers of the spinal cord, and they show lots of interactions between different classes of afferent neurons shown by different colors. And these are mediated by lots of different sorts of interneurons. So little is known about these deep interneurons that modulate outputs that convey touch information to higher brain centers. We do know that in the spinal cord, you get interactions between touch and pain. And this is why if you hurt yourself, you hold onto your hand really tight. It's because there's a inhibitory interaction from touch to pain. It makes it less painful. Also, touch with temperature and even touch with touch. So most of these interactions are inhibitory in the spinal cord. There are probably lots more interactions in the brain where they might well be excitatory, but at least in the cord, they're inhibitory. This is really important in terms of perception, because it means that if you have a complex stimulus, for example, one which has touch and temperature, slow touch and fast touch, touch and pain, then what reaches the brain is not simply a linear superposition of the component stimuli. It's not that all of the receptors just project independently to the brain, which makes a linear sum, because there are interactions in the spinal cord. And this is an area which is kind of not really well understood and quite hard to study, but probably really important. Okay, I've got a couple of slides which are more about perception and less about neuroscience now. I've called this cognitive architecture of touch perception. So we're familiar with the idea that we can perceive objects with our hands. If it's dark, I can find objects on my desk just by using my hands. I can tell if it's dark and I'm coming into my house, here are my keys. I can use touch to distinguish which is my bicycle key, which is the top front door key, and which is the bottom front door key. I can do all of that just by tactile sensation, by touch perception. So how does that work? And how do I get the sensation, oh, that's the bike key, or that's the front door key? So again, it's worth thinking about the pathway. And here is just another example of the information coming in through the neuron into the spinal cord, up into the brainstem, and into the primary somatosensory cortex, which we've just seen. Now, we don't know much about the somatosensory pathway in the brain after the primary somatosensory cortex. So we know quite a lot up to the primary somatosensory cortex, but very little is known about what happens after that. What we do know is that there's a continuation of the pathway backwards from the primary somatosensory cortex towards an area called the posterior parietal cortex. And the posterior parietal cortex is an area where all the different sensory modalities seem to come together. You get visual information, auditory information, tactile information, muscular information, and somehow the posterior parietal cortex seems to be the place that the brain puts together all of its inputs to produce a representation of what is the object. You get a transition from what we call sensation to what we call perception. And in terms of touch, that has two aspects. The first one is as you go higher and higher and higher in the somatosensory pathway, beyond the primary somatosensory cortex S1 and into the parietal cortex, you get a progressive integration across receptor modalities. So you remember that there are four main classes of mechanoreceptor. There are also muscular receptors. And as you go farther and farther back, what you find is neurons which will respond either to touch on the skin or to the stretch, the passive stretch of a muscle when it's moved. So the actual mechanical stimulus at the receptor becomes less and less important. And the neurons seem to code what is the external physical event which is causing the receptor to fire. So you get what might be called integration across receptor modalities. You could also call it abstraction from the kind of details of the stimulation to something which is more like the actual object that is causing the stimulation. Posterior parietal cortex is basically doing what computational people call causal inference. What is that thing touching me? Another really interesting thing that happens as you go backwards from primary somatosensory is that you get a progressive loss of somatotopy. The receptive field sizes, in other words, the bit of the skin that a given neuron will respond to, gets bigger and bigger and bigger. So in primary somatosensory cortex, you get neurons which will respond just to a tiny patch on one fingertip. As you go back, you'll get neurons which will respond to a stimulus anywhere on the hand, anywhere on the body, and so on. And also, you interestingly lose this disproportion. So here's the somatotopy in the primary somatosensory cortex, big lips, big hands. And as you go back, I'm afraid I can't easily read the labels here. This is in the medial wall of the parietal cortex, and this is in the insula. You'll see that this little man has now got bigger legs. He's got a head which is more in proportion to the rest of his body, and his hands have grown relatively smaller. So that's just a visual representation of the fact that as you go higher and higher up the somatosensory pathway, you lose the topographical features that reflect the receptors, and you start to see topographical features which reflect the proportions of the way our physical body really is. And that's why we think that the higher parts of the somatosensory pathway may play a particular role in bodily representation, bodily awareness, and self-awareness, understanding what this Patrick thing, this sensory body is like, not just sensing stimuli, but having an awareness of the self as well. And a final thing that we notice higher up in the somatosensory pathways, we get a lot of bimanual interactions. So the corpus callosum, which is the thick bundle of neurons that connects my left hemisphere with my right, has many, many fibers at parietal level, which convey tactile information from one side to the next. And many, many neurons in the parietal cortex will respond exactly the same way, whether the right hand is stimulated or whether the left hand is stimulated with the same stimulus. So there's something about sort of integration from across the whole of the body. The second thing I want to say about cognition is that tactile bandwidth is low. This is something which all virtual reality engineers should be made to repeat 100 times every morning. Tactile bandwidth is low. Here is an experiment by Charles Spence where they put tactile stimulators all over your body, and they stimulated you with, these are little vibrations. They stimulated you with many vibrations at a time, and they asked you how many touches can you feel. So it could be 1 or 2 or 3 or 4 or 5 or 6. And here is the number of stimuli that happen in parallel simultaneously. So this is the number of buzzers that you get. And this is the number of errors that you make, okay? So when you deliver 2, 3, 4, 5, 6 stimuli in parallel, people never say that was 2, that was 3, that was 4, that was 5, that was 6. What they tend to say is, I don't know, loads, maybe 2. And above about 2, people just don't know how many tactile stimuli they're getting simultaneously. Basically, you can only really perceive one tactile stimulus at a time in a proper sort of signal detection type of paradigm. So this is really odd because, you know, at one level, we're aware of the touch of our clothes on our body constantly. But at the same time, if you deliver multiple touches all at the same time, people are what is called simultanagnosic. People are unable to perceive simultaneous stimuli. And this, of course, makes a real challenge for all the traditional engineering approaches which have aimed at tactile TV. So this is one of the stranger images in tactile neuroscience. I'm afraid I couldn't resist downloading it simply because it's so bizarre. I hope nobody objects to it. And here, this person has an array of tactile stimulators on their back, which are connected to cameras in their glasses. So when they look at an object, a set of tactile pixels on their back will translate the visual object into a tactile image, which is projected onto their back. And this work by Bakirita was basically disastrous because they spent loads of money on equipment and all they could really do is distinguish a face from a telephone. So the bandwidth is really too low. In addition to the bandwidth being low, there aren't actually many tactile receptors on the back anyway. So subsequent attempts at multi-touch stimulation have tended to use areas like the tongue. So this is the same idea, and it's still pretty hard. It's still pretty hard to get any kind of spatial percept out. And that's led some people to say, is touch actually a spatial sense at all? Maybe it's not a spatial sense. And this is the only piece of my own work that I'm showing today. In fact, we think that touch is a spatial sense. So here what we did is we presented a pair of these two white stimuli, which basically define a line. And then we presented one of these gray, these are tactile stimuli, which are normal to the skin. They're little solenoids. We presented another tactile stimulus, and you have to say whether the gray stimulus is on the right or the left of the line defined by the previous two stimuli. And this is your probability of saying the second stimulus was on the right of the line defined by the first two stimuli as a function of where the second stimulus actually was. And people can do this, people can do it reasonably well, and it shows that they must represent the line. People are able to represent when these stimuli are all lined up. So touch must be a spatial sense, but only in this rather crude sense of being able to detect a line of three tactile stimuli separated by a few centimeters. So you can't get really high resolution parallel stimulation through touch. And instead, a lot of the interest in tactile engineering has gone on temporal information. So you may remember I showed you these rotating drum type stimuli where you can present a texture of dots to the fingertip and you can rotate a cylinder which you can touch with your fingertip. So then what you get is only one stimulus at a time, but many, many stimuli in a time series in succession. And that's perhaps what you need to do when the bandwidth is low. Okay, so my final point, called active touch, is actually a joke point because we're running out of time and it's in quote marks. So a lot of people talk about active touch and it's really, it doesn't exist. So when you exactly control to make the stimulation identical between active touch of an object and passive touch of an object, perception is the same. People are just as good at perceiving touch when it's presented passively to their skin as when they actively touch. The critical thing is you've got to make the stimulation exactly identical. In active touch, what I do is I explore the object to get the most informative stimuli I possibly can. I'm looking for information and I'm getting great information. If you build a tactile stimulator which gives the same information to the passive hand, people will have just as high a level of performance. So most of what you read about active touch is showing superiority of active touch is actually not good neuroscience because they haven't made the stimulus identical in the active and the passive condition. So I can go straight to my last slide, which is, it was supposed to be conclusions, but actually I've tried to write it more as some issues for developers. Suppose you want to use all this knowledge because you're an engineer and you're trying to build a tactile interface. Here are just some things you need to think about. What receptors will my stimulus target? Okay, what are the receptors in the body, in the skin or in the muscles or wherever, probably the skin? Which kind of receptors are they? Are they rapidly adapting, fast adapting? How many of those receptors are there? If you're stimulating people on the back, there aren't many receptors for your stimulus to target. If you're stimulating them on the lips, there are going to be loads. Be realistic about the communication bandwidth. How much information are you trying to send? And remember that your bit rate is going to be quite low. You can't send many tactile stimuli at the same time. You can't multiplex. You can't send parallel streams. A really important thing is to choose which skin region you're going to stimulate. So choice of body part is a really important first step because it involves lots of trade-offs. Some body parts are difficult to get to because they're under the clothing. Some body parts are very, very sensitive because they have lots of receptors, fingertips and lips, but also feasibility. So if you have the fingers enclosed in a tactile stimulator, then people are not going to be able to do lots of other things that they want to do with their fingers, like scratch their face or gesture with their hands. So pick carefully where you're going to put your stimulator on the human body. And then the final thing, which I've learned from a number of, sometimes funny, sometimes unpleasant experiences in my own research career, is you can have a lot of fun with tactile stimuli. Look at the facial expression of people while you deliver them a tactile stimulus, and they're often smiling, but you've got to be careful. It's quite easy to hurt people unintentionally, so you need to be really good at risk management. There are physical risks, and there are also really important sociocultural risks. Almost all human societies very carefully regulate who can touch your body, and there are lots of good reasons for that. So the risks are real. So that's it, and I'm going to stop there and stop sharing my screen, and I can stay for at least 15 minutes to answer any questions. Thank you very much.
Thank you, Patrick.
I think I'm going to stop recording the Q&A just in case people feel that they'd rather not be recorded. That will encourage questions. So I think I just press there and